I'm going to do a chemistry experiment where I use red dye 40 found in strawberry Kool-Aid to dye different types of fibers. And these cloths here have 8 different fibers on them, and the names of those fibers are acetate, cotton, nylon, polyester, polyacrylic, sometimes just called acrylic fiber, silk, rayon, and wool. I have 3 different cups here, beakers, and in the first beaker there's just water, in the second beaker there's vinegar, and in the third beaker there's hot vinegar. I'm going to add a little bit of the strawberry Kool-Aid to each, and all I'm trying to get from the strawberry Kool-Aid is some red dye 40. Try to get the color fairly uniform in each to get a similar concentration. So Kool-Aid has citric acid in it, and so all of these beakers have had citric acid to them. This one here, which just has water in it, is now going to be slightly acidic from the presence of citric acid. This beaker here has vinegar in it, and vinegar is in there to actually keep the pH at an acidic pH 2, and this is hot vinegar, so the pH there is also probably approximately pH 2 as well. I want this beaker to be basic, so in order to do that I'm going to have to neutralize the citric acid. And to neutralize the citric acid, I'm just going to add a little bit of baking soda. So it's going to probably form some carbon dioxide gas. So this is going to foam a little bit as the citric acid is neutralized. And this is going to take a little while. I want to continue to add baking soda until the foaming stops. And then once the foaming stops, I'm going to continue to add baking soda so that the pH will be that of an approximately a baking soda solution. So that'll be a pH of about 8 or so. And I think we have probably neutralized all of the citric acid by this point. To summarize, this is going to have red dye 40 at a pH of about 8. This is going to have red dye 40 at a pH of about 2, and this is going to have hot red dye 40 at a pH 2. Now I'm going to go ahead and add my 8 fiber cloth to each beaker. I'm going to let that set for about a minute. Now that 1 minute's up, I'm going to go ahead and remove the fibers from each of the beakers, and I'm going to give them a good rinse in some soapy water. And I'm going to go ahead and rub the fibers so as to remove any dye that's not really adhered. So I've removed it from the dye bath at pH 9. We'll remove it from the dye bath at pH 2. Give that a rinse as well. And we'll also remove it from the dye bath at pH 2 that was hot. Let's go ahead and analyze our results. The fibers have been removed from the dye baths. We see over here on the left some control fibers that were untreated. Here are the fibers that were treated with the red dye at pH 8. Here are the fibers that were treated at pH 2. And here are the fibers that were treated at pH 2 at a high temperature. To understand the binding behavior of the dye to the fibers under the various conditions, we're going to need to think about the intermolecular forces between the dye molecule and the various fiber molecules. First, let's take a look at the chemical structure of the dye, red dye number 40, also known as Allura Red. This will help us gain some insight into the binding behavior observed. Red dye number 40 contains two sulfonate groups. These are strong acid groups, and so they're going to be deprotonated and carry a negative charge at all pHs studied in our experiment. These sulfonate groups are going to be able to participate in ion-ion binding, and they're also going to be able to participate in ion-dipole binding. There's also this hydroxyl group here that will be available to participate in hydrogen bonding. Now remember that this dye is dissolved in water, and this dye is going to be strongly associated with water because obviously water can hydrogen bond to this hydroxyl group, and there's going to be strong interaction between water molecules and these sulfonate groups through ion-dipole forces. So in order to get this dye molecule to bind to a fiber, the fiber-dye association is going to have to be fairly strong because the dye is pretty much going to want to stay dissolved in the water. In an equation, I sort of see this as the dissolved dye molecule will react with some sort of fiber molecule to form a dye-fiber association. If you want to dye a fiber, you're going to want to drive this reaction as far as you can to the right, so you're going to want to have strong intermolecular forces between the dye and the fiber molecules. The first two results I want to take a look at are those of cotton and rayon. Now here's our cotton band here, and here's our rayon band here. And in every single case, it looks like the fiber, in both cases, both cotton and rayon, are dyed a pink color. And the strength of the pink color doesn't seem to change much with pH. Now these results make sense if we recognize that both cotton and rayon are made of cellulose, the chemical structure of which is shown up here at the top. Cellulose is a bunch of glucose monitors linked together in a polymer. And as we inspect the cellulose polymer chain, we see a lot of hydroxyl groups. And those hydroxyl groups are not going to deprotonate on the basis of pH. So at pH 8 and pH 2, the chemical structure of this is going to look the same. Now, because of these hydroxyl groups on the cellulose and the hydroxyl group here, there's going to be hydrogen bonding that's going to be available between the Allura Red, the red dye number 40, and the cellulose chain. There's also going to be able to be the presence of some ion-dipole forces between these hydroxyl groups and the sulfonate groups on the red dye number 40. So this dye molecule will bind fairly strongly to cellulose, which is both cotton and rayon in every case. And because the dye molecule does not change its structure on the basis of pH, and because the cellulose backbone doesn't change its structure on the basis of pH, we wouldn't expect any change in the type of or the strength of binding when the pH changes. And that fits our observations pretty well. As we look at the cotton band and the rayon band, we get pink fibers in almost every case. Now we're going to analyze the acetate band. That's this band here at the top. And we see the acetate band does not dye very well. We can understand this if we recognize that acetate is made of cellulose acetate, the structure of which is shown here at the top. Now, cellulose acetate is very similar to cellulose, with the exception that many of the hydroxyl groups have been esterified. And when these hydroxyl groups are esterified, this limits the ability of our dye molecule to hydrogen bond to the hydroxyl groups on the glucose because, well, they're no longer there. This is also going to limit the strong ion-dipole forces that can exist between the cellulose acetate and the dye molecule. So we would expect weaker bonding between the cellulose acetate and the dye, and of course that's what we see. We don't see any dyeing of the acetate band in our experiment, And that just means that the dye molecule probably prefers to stay dissolved in water because of the weakened association with the cellulose acetate due to the esterification of the hydroxyl groups. Now let's take a look at the polyester band. Like acetate, polyester does not dye very well with red dye number 40. And if you look at the monomer of polyester, so this is a monomer unit of polyester. There's many of these linked together to make polyester. If we look at this structure, we don't see a whole lot of opportunity for the dye molecule to bind to the polyester monomer. It's because we basically got a lot of carbon and hydrogen bond, and we've got some ester groups here that are going to form weak associations with the dye. So of course we're going to expect that the polyester does not dye very well, and that's exactly what we see. Similar in the acrylic fiber, or polyacrylic, we also don't see any binding of the dye. If we look at the structure of polyacrylonitrile, which makes polyacrylic, we see a lot of carbon-hydrogen bonds and carbon-carbon bonds, which are not going to be able to bind to the dye very well. See an ester group here that we've talked about before, doesn't bind to our dye molecule very well. We also see these cyano groups. Now, a student might be forgiven if they say, you know, the hydrogen on this hydroxyl group on the dye could probably hydrogen bond to this nitrogen on the cyano group. Well, in actuality, these cyano groups are more like pseudo-halogens. So they act more like chlorine atoms or bromine atoms or iodine atoms connected to the carbon backbone. And we know that chlorine, bromine, and iodine aren't going to hydrogen bond to this hydrogen here. And because these cyano groups act like those halogens, there's not going to be any hydrogen bonding that's going to occur between this hydrogen on the hydroxyl and the cyano groups. So there's going to be a weak association between the acrylic fiber, the polyacrylonitrile, and the dye molecule, and that bears out in our experimental result. We see no binding of the dye in the polyacrylic band in any case. We're now going to turn our attention to the protein bands. So silk and wool, those fibers are made of protein. Silk is made of fibrin protein and wool is made of keratin protein. And to understand the results of the proteins, we're going to need to think a little bit about protein structure. So proteins are made of amino acids linked together, and I've tried to designate that here. This is a single amino acid here, connected to another amino acid here. Looks like I have, how many? It looks like about 6 of them here in the chain. This is a basic amino acid side chain. This is an acidic amino acid side chain. And this would be the case if our amino acid chain was at a low pH. In that case, our basic side chain is going to be protonated, and our acid side chain is going to be protonated. And if you look at the chain as a whole, we see that the chain contains a positive charge. And that's generally true. At a low pH, a protein is going to be positively charged. Now, if that protein is at a high pH, then the base is going to lose its proton, becoming neutral. The acid is going to lose its proton, becoming negative, and so we recognize at a high pH, proteins are going to be negatively charged. Now, every protein is going to have a point of pH where they're neutral. So the pH at which a protein is basically neutral, that's called the isoelectric point, or the PI for short. And it's good to be thinking about this when we look at the results for the dyeing of wool and the dyeing of silk. So let's check that out here on this slide. I see the silk band. I don't see much evidence of binding of silk at pH 8 or of wool at pH 8. But when that pH is lowered to 2, I do see evidence of binding in silk. And I see some slight evidence of binding here at room temperature at pH 2 for wool. And I see good evidence of binding at pH 2 for wool when the temperature is raised. Now this makes sense if we remember that at a low pH, like pH 2, our protein is going to be positively charged, And boy, these sulfonate groups here are really going to like that positive charge on the protein. So the dye is going to bind to the protein fibers really well at low pH. And that's what we see. We see the dye associated with both silk and wool at a low pH. However, at the higher pH, we would expect the protein to be negative. And of course, a negative protein is not going to want to interact with these sulfonate groups. Negatives repel each other. And so we would not expect to see much association of the dye with the silk or the wool at a high pH, a pH 8. And that is indeed what we observe. I'd like to talk a little bit about this result where wool does not seem to bind very well at room temperature at pH 2, but really well at the same pH at an increased temperature. To think about this, we're going to imagine a single strand of protein of wool, sort of like this curved line of spheres here. Now each sphere is going to represent an individual amino acid. And along this single strand, there's going to be interactions, hydrogen bonding interactions, that are going to force the wool protein strand into this sort of helical shape. It's actually an alpha helical structure in wool. Now these associations are hydrogen bonding associations, and they cause the wool strand to be very tightly wound, and these associations are very strong. In addition to these intra-strand associations, there's also inter-strand associations. So there's strand-to-strand associations. So one alpha helix of a wool strand can interact with a separate protein strand through protein-protein interactions that are strong as well. So for our dye molecule to be able to bind to these various strands, it's going to have to overcome some pretty strong interactions. That's difficult to do even if the protein is positively charged at room temperature. However, if the temperature is raised, that thermal energy can cause these associations to break and to weaken, causing the helical structure maybe to unwind a little bit and these strand-to-strand associations to weaken or even break. If that happens, binding sites for the dye are going to open up on these strands, and it's going to allow the dye to bind to the protein strand. So we'd expect better binding at a higher temperature under these circumstances, and that's what we observe with wool. Finally, we're going to look at the case of nylon, which in fact shows some binding characteristics that are similar to wool. At pH 8, we see almost no binding. We see some fair binding at room temperature at pH 2, and some strong binding at pH 2 at an increased temperature. Up here on the upper right, we see the chemical structure of nylon. Like proteins, it's comprised of a polyamide chain. So here's a top polyamide chain. And this polyamide chain is going to be associated with other chains through hydrogen bonding interactions. Much like proteins, nylon has a pi, an isoelectric point. That happens to be around 3.9 for nylon. So below a pH of 3.9, we would expect nylon to have a positive charge. That happens on these terminal nitrogens. They become protonated. Below a pH of about 3.9, these start to take on positive charge. And so at a pH of 2, we'd expect nylon to be positively charged, and we'd expect a strong association between the positive charges on the nylon and the sulfonate groups on the red dye number 40. Now, if we could increase the temperature, then these interstrand associations would weaken. The thermal energy would go into breaking these hydrogen bonds. And if the strands were to dissociate from one another, that would open up binding sites wherein the dye molecule could attack the various strands and undergo hydrogen bonding and dipole ion forces. And so the dye molecule is going to be expected to be able to bind to nylon more strongly at a higher temperature. And if we look at the nylon band, indeed, that's what we see. Nylon binds much better at a higher temperature at pH 2 than it does at a lower temperature at pH 2. So the results for nylon and wool are quite similar. So hopefully by watching this video, you've learned a little bit about the complicated chemistry that's involved in the dyeing process. I hope you do some of your own experiments with some food dyes or other Kool-Aid dyes and different fibers. Enjoy your experimenting.